Hi everyone. I have had to make a change. It seems that the kit for Lab 13, um, I can't even remember what it was called, but Lab 13 that goes along with your Chapter 15 on genes and proteins, um, the kit does not include the correct beads, so you can't do it. And the last part of it, the DNA extraction, you already did that in the previous chapter. So I want to show you how to do this virtual lab. This is a virtual lab that will go over some of the concepts, and it will especially help you understand what happens when there are DNA mutations, what actually happens in the protein. Because hopefully by now you understand that the genes are pieces of DNA that code for specific proteins. So, where we've been saying DNA codes for our traits, now we can say DNA codes for protein. And the first step in that process is called transcription. That is where DNA makes a copy of the gene called messenger RNA. And then the second step is called translation, and that is where the messenger RNA codes for a specific protein. And that means it has to code for the specific amino acids in the protein that form the protein. Okay, so we're going to click on first the lab report so you can see what it looks like. There are two ways that you can complete this lab report. You can either print it out and do it by hand, or you can do screenshots and replace this picture here. You can replace this, just delete this picture and replace it with a screenshot of the original sequence filled in. But let's look at the lab first. Okay, so when you click on the link to the lab, go to the assignment, and here's the link to the actual lab activity. Okay, so when you click on this, your instructions are over on the left. I'm not going to read them because you can do that. And then I strongly encourage you to read through this mutation guide. Um, just go through and read through it. It explains the two different types of mutations: a silent mutation, which actually doesn't result in a change in the protein. It's a mutation in the DNA sequence that actually does not result in a change in the protein. It doesn't have an effect on the protein at all. And then there's a frame shift mutation. Um, and then there are what are called point mutations. And I do not see. Oh, I'm sorry, I skipped that one. Okay, so there's your mutation guide, and you need to read it. And then here is your amino acid chart, and I'm going to show you how to use that in just a minute. Okay, so let's look at the worksheet again. Now, this worksheet has a specific example. It's got the mutated messenger RNA and amino acid sequences listed. And the amino acid sequences are, I'm just going to look at the first few: methionine, serine, and I think it's glutamine or glucine. Anyway, no, it's glutamine. Anyway, you don't have to know that. This is the abbreviation for the amino acid name. If you want to know the amino acid name, there's a chart that shows you. But right now, I just want you to remember methionine, serine, and GLU, whatever that stands for. We're going to come back to our lab, and we are going to hit reset until we see that. Hit reset and mutate until we see that methionine, serine, and GLU, I can tell you now, is glutamate or glutamic acid or glutamate. So this is the sequence that you see on the worksheet. Now, I know that's a little bit tedious, but that's what you have to do. You have to actually find this sequence, and then you have to come in here and you have to complete it. Okay, so you know if the rule down here, it says mutation rules, it says the mutation rules are insert A in front of second G. So in the original sequence, the second G has an A inserted in front of it. So in the original sequence, and I'm going to go ahead and do this one for you, you're going to have the same thing as the mutated sequence, except if the mutated sequence has an A inserted in front of the second G, then this A is not there in the original sequence. So now I'm just going to skip that A and follow the rest exactly like it is in the mutated sequence. This takes a little bit of time. That's okay. It's just click and drag. You guys are probably faster than I am at this. I feel like I need to talk because it's boring, but the more I talk, the more mistakes I make. And I'm trying to do this as fast as I can. So you simply go back to your original strand, look at your mutation rule, and figure out what the original strand must have been, what the sequence must have been. Okay, so that's what it would be. Now, I already know that AUG, every 3 nucleotides in your messenger RNA, is called a codon, and a codon codes for a particular amino acid. I can already see that AUG codes for methionine, MET, methionine. So all I have to do is find that. Actually, this is called your start codon. Then I've got a GCG, and I don't know what that is, so I'm going to look at my chart. Here's G, and then here's the column for the C's, and GCG is alanine. So I'm going to find alanine and put that here in the second box. AAC, asparagine. You see it? AAC, asparagine. CAC is histidine. GCG is alanine. And then CAC, didn't we have that before? Yes, histidine. ACC, threonine. And then CUG is leucine. And as you can see, the sequence of amino acids in the original sequence, after you pass methionine, it is completely different. And when the amino acid sequence is different, then that means the protein is no longer the same protein. And so that can cause a mutation in your in the trait that the protein is responsible for. So I'm going to click on check. Let me get off of mute so you can hear it. Okay, check. Yay, I'm so glad I got that right, because it's really easy to make a mistake. But this is how you do it. And now you're going back to your worksheet and you have either handwritten the answer on your printed sheet of paper, or you come here and you delete this and you come back to this page, you hit print screen. On my computer, there's a little window symbol and then a PRTSC button. But if you can print your screen or do a screenshot, I can't tell you how to do that on every computer, but I just know I can do it on mine, and so I'm going to paste it. And then I don't want all of that, so what I'm going to do now is crop, and I'm just going to post this part. Okay, and then I can make it bigger, which I hope you will do. And then you're going to answer these 4 questions, and you're going to go on to the next example. So then you'll save it, and then you will upload it. You will upload it to, I don't know what I did, I'm sorry.